Making pico de gallo, huh?
Oh, yeah.
I guess. I don't know. I need my garlic
Oh,
sauce.
shit.
Oh, what the?
Oh, hey, I didn't see you there.
Ooh.
My name's Santino.
And my name's Abar. Oh, so you guys are trying to do the 2026 FRQ?
We'll teach you how to cook this
Yeah,
up.
we'll teach you how to cook this. Let's head over to the gazebo.
All right, now let's get started on the first part of our question. I'm going to pause the video now and read the question on screen for part A and press play when you're ready.
Since the prompt uses the verb describe, we can't just list our answer. We have to actually provide some detail about this question. This scary sounding molecule called dinucleotide polyphosphate may seem hard to understand, but the question literally tells us it's just a type of nucleotide. Santino, do you know how to solve this question?
Me? I don't know. I literally forgot all of that. That's like a year ago. How about you handle this question?
I think I also look forgot what the three parts of a nucleotide are. So,
How do we solve this? How do we do this?
oh, we should call in the great Mr. McDaniel for help.
McDaniel? McD?
Are
McD?
you ready?
I'm so ready.
Oh, great dolphin
great
lord.
dolphin
Oh,
lord.
great dolphin
Oh,
lord.
great.
Zawater!
Dear pupils, why have you summoned thyself? So, recall from Unit 1 and Unit 6 that a nucleotide consists of three major parts. It includes the phosphate base, it includes the nitrogen base, and don't forget that nucleotides also include a 5-carbon sugar. It'll be deoxyribose for DNA and ribose for a RNA. Now, take your leave. I will see you again.
Thank you so much. So I guess our answer is a nitrogenous base, a 5-carbon sugar, and a phosphate group.
Wait. Be careful, Santino. You just identified the three part, but you didn't really describe it like the question asks. Instead, you should probably elaborate more on the function of each of these three structural parts. Can you try?
So like, man. Well, the nitrogen base is responsible for acting as a coding part, such as adenine or thymine, and the 5-carbon sugar is a central piece that connects the phosphate group and the nitrogenous base. Then the phosphate group is a part that provides structural support by holding the molecular backbone together.
There you go. Easy question.
Wow, this text is pretty lengthy. Go ahead and pause the video and read it. I'll skip the text.
So we know from the text that AP4A and CP4C bind to DORN1 receptors on the surface of the plasma membrane, which triggers a cascade of ATP and signaling molecules. In this case, the effect of this signaling cascade is a stomata closing. And that's actually represented in the graph of the effects of the treatment, the stomatal size. AP4A, CP4C, and ABA treatments have some of the smallest stomata.
If you look at that, it's a bar graph. So funny! Hey, Bart, tell me about it.
Well, there is one thing to consider when analyzing a bar graph, especially in AP Bio. It's basically all about variables. First, these scientific graphs that display data are usually structured to have the dependent variable as the y-axis and the independent variable as the x-axis.
Well, what the heck is an independent and dependent variable?
Well, in simple terms, an independent variable is the cause, which is basically the part of the experiment that you change, and the dependent variable is the effect, the part of the experiment that you observe or measure. In this case, the independent variable is the different chemical treatments on the x-axis, while the dependent variable is the size of the stomata opening on the y-axis.
Now using that information, we can verify the dependent variable in the scientist's first experiment is the size of the stomata opening. So it is both on the y-axis and it's what the scientist is measuring.
That's right, but don't forget, graph positions don't always show that something is dependent or independent, but it is usually a cheat code to identify what the variable is.
So now to solve part 2 of B, we simply need to know how to read a graph. We're asked to describe the relative size of the stomata in CPC treatment group as compared to the stomata size in the buffer group, both of which information is given by the graph. Based on the graph, we can see that CPC stomata size is about 0.6 as compared to the buffer stomata size of 1. However, a proper answer in AP Bio will require the usage of an important phrase called insignificant or significant difference. It has to do with the error bars of the graph.
Oh, you must be talking about those weird lines on the bar. Can you explain what that means statistically?
Great question. The two lines that are sitting on the top and bottom of the graph are known as error bars, which essentially represent something known as the standard error of the mean, SEM for short. If the error bars between the group don't overlap, for example, our treatment group versus our buffer group control group, that means there is a significant difference between the two groups.
Oh, so in this case, since the CP4C error bar is not overlapping with the buffer error bar, there is a significant difference.
That's right, easy points.
Oh, so now we just need to justify why we use a buffer as a control group. I would say this is a key feature of running a scientific experiment. So basically, the buffer acts like a baseline for us, and it shows us the normal conditions of the stomata when there's no added signaling molecules. Basically, we can confirm that any changes that does happen in the stomata is due to some other active signaling molecule. Oh, so now for part C. Reading the question, it looks like we're introduced to a second experiment, where basically everything is repeated except the DORN1 receptors are mutated.
These mutations are essentially accidental changes in DNA sequences, resulting in a change in where protein is usually produced. This change in the protein can dramatically shift the function for a specific cellular mechanism and how it works.
Right. Now let's get to solving part 1 of C.
And read the question. As you can see, APA in the graph is used as a positive control variable, where a variable is used to get an expected outcome, so it is given that this molecule causes a stomata to close.
Okay, well, what about part 2 of C?
So hopefully you read that question. APA reacts to the DORN1 mutation by not being significantly smaller to the buffer, while CPC still makes the stomata smaller in the presence of the DORN1 mutation.
Okay, and what about part 3 of C?
Read the question again. We know that the DORN1 receptor makes plant cells create molecules that cause the stomata to close. Given by the graph of figure 2, APA is shown to not close as much compared to the buffer, showing the fact that there is a significant decline of the molecules produced compared to the non-mutated DORN1 receptors shown in figure 1.
So it looks like this mutation is just causing the intracellular domain, which is basically the inside of the DORN1 receptor, to be missing.
Let's look at 1 and 2 together. From this intro, this definitely ties into Unit 6, gene expression. Just some background info, spherical molecules usually are able to land on a receptor, which in turn causes the receptor inside the part to change shape. That change in shape is crucial as it allows for a message to be passed down into the cell all the way into the nucleus, where specific genes are turned on. Since the inside of DOR1 is gone, the message won't go through the nucleus, therefore no transcription of the plant defense gene in the mutation cells.
Oh, well, you basically just answered part 1. So, moving on to question part 2. How can you justify this?
We can explain that without the intracellular domain, there is no signal transduction pathway, so no transcription factors are able to be turned on to activate specific defense genes.
And that's the last question.
Just like that, by breaking down the text, tracking the independent and dependent variables, and leaning on our basic unit principles, we walk through the entire long-form FRQ step by step.
Listen, don't let the heavy vocabulary scare you. Look at your error bars, think about your control groups, use your analogies, and you'll do great.
Thanks for watching, guys. Good luck studying.
Noise pollution.